Hi, thank you for taking the time to view this presentation. During the course of this project, my partners and I realized that we all shared something fairly interesting in common, that we love Brussels sprouts. We had a lot of fun working on this presentation, and we're happy that you are here to view it. This presentation is called Continuous Production of Lentiviral Vectors for Gene Therapy. Before we get into the meat of the presentation, we're going to be going over a brief abstract to get an overview of what was accomplished during this project. The aim of this project was to design a lentiviral vector production facility capable of producing 1 × 10 to the 14th active virus particles per year to enable a price reduction in expensive gene therapies while still maintaining profitability. The method used was SuperPro Designer, and it was used to construct a rigorous model of a mostly continuous lentiviral vector production facility. The results obtained were a production of at least 1 × 10 to the 14th virus particles per year at titers of 5.79 × 10 to the 12th, while running at 50% overproduction for contingency purposes. The upstream productivity was 1.6 × 10 to the 13th virus particles per week. The downstream efficiency was 35 to 40% yield. The fixed capital investment was $44.9 million, and the annual operating cost was $17.9 million. Next, we're going to be looking at a brief introduction to see how this project played into the larger industry as a whole. Gene therapy is a valuable industry. It has a higher expected 7-year compound annual growth rate than the rest of the pharmaceutical industry, at 16.6% versus 7.4% respectively. Now, this has the effect of gene therapy taking an increasingly large part of the pharma market revenue, from 2.8% in 2019 to 5.4% in 2027. This is because gene therapy has very expensive drugs. With prices ranging from $373,000 to $2.1 million, it's no surprise that a 2019 survey of U.S. gene therapy payers found that 80% were highly or extremely highly concerned with the cost of their drugs. Furthermore, another 2019 survey of U.S. citizens at large found that 88% desired to increase the generic drug market, and 63% desired to increase taxes on companies with expensive drugs. Now, these could both have negative consequences for large companies producing gene therapy. In order to satisfy consumers while still maintaining profitability on the drugs, these drug companies need to reduce their cost of production. One way of doing this is the method proposed by our group, which is the continuous manufacturing of lentivirus to reduce waste. The National Science and Technology Council estimates that continuous manufacturing can save 40 to 50% of variable cost of production. Lentivirus is a good candidate to use continuous manufacturing with because it is one of the most common viral vectors used in gene therapy. It has demonstrated safe gene transfer in T cells and stem cells, and it is able to address current markets, which are dominated by ex vivo CAR T therapies, and future markets, which are expected to be dominated by in vivo precision medicine. Next, we're going to be moving into the process description and innovations relative to the platform process, starting with the upstream process. The upstream process can be broken down into 3 production sections: inoculum preparation, cell propagation and concentration, and lentivirus production and recovery. 2 important considerations that needed to be made for the upstream process were the choice of cell line and media. For the cell line, a HEC 293 cell line was chosen. Specifically, this cell line had to be stable producing, which enabled us to use long production cycles, capable of suspension growth, which enabled scale up and not out, capable of serum-free growth, which reduced costs and media variability, and finally, it needed to be capable of induced production. The media that we chose was CD293 AGT media. Inoculum preparation was the first phase of the upstream production. The equipment used for this was a 2-liter and a 50-liter Biostat RM single-use rocking bioreactor, as well as a Repligen X-Cell ATF alternating TFF. This was used to retain cells for the 50-liter perfusion. The goals of the inoculum preparation were to minimize necessary equipment and to quickly propagate the cells. The novel strategies employed were using high cell density 5 ml cryovials to directly inoculate the 2-liter reactor, and also using batch fermentation to obtain proper cell density for perfusion and then using perfusion to further increase the cell density. The result obtained was 50 liters of 2 times 10 to the 10th cells per liter in 9.5 days. The next phase of the upstream was the cell propagation and concentration. The equipment used for this stage was a 500-liter working volume high-performa Dynadrive single-use bioreactor and a Repligen XL ATF-10, and this was used to retain cells during perfusion. The goals of this stage were to quickly and effectively generate 500 liters of high-density cell culture, 475 of which will be used for virus propagation, and 25 will remain in this bioreactor for re-inoculation. The novel strategy employed was using high turndown ratio single-use bioreactors for 10 weekly cycles before their disposal. The result obtained was 500 liters of 6 times 10 to the 5th cells per liter obtained in 5 days. The last stage of the upstream was lentivirus production and recovery. The equipment used was a 1,000-liter stainless steel Sysbiotech bioreactor, a Repligen XL ATF-10, and a cross-flow KTF TFF. This was used to remove virus particles during perfusion. The goals of this stage were to produce and isolate virus particles for a 5-day period, and also to reduce costs by recycling media. The novel strategy employed was using a 2-filter system to isolate virus particles and recycling 2/3 of the 60 liter per hour of perfusion media. The result obtained was 1.6 times 10 to the 13th virus particles produced per week. Now we're going to be moving on to the downstream. The downstream involves 5 separation sections: clarification, purification, DNA removal, concentration and formulation, and sterilization. The first downstream step, clarification, used a 0.5 micron TFF. The goals of this stage were to continuously remove cell debris and continuously recover high percentages of virus particles. The novel strategy employed was using continuous TFF as opposed to centrifugation, which resulted in large product loss, and diafiltration, which only operates in batch mode. The result obtained was greater than 99% of product recovery. The next stage in the downstream was purification. The equipment used for this stage was 3 separate 4,000 milliliter CIM DEAE weak ion exchange columns from BIA separations. The goal of this stage was to recover virus particles as continuously as possible. The novel strategy employed was sequential column chromatography to enable continuous input and semi-continuous output of product stream. The result obtained was 80% product recovery. The next stage was DNA removal. The equipment used for this stage was a batch bioreactor. The goal was quick and cost-effective removal of host cell DNA fragments using benzonase. The novel strategies used were performing the reaction at 37 degrees Celsius for 15 to 30 minutes rather than 4 degrees Celsius for a period of a night, and performing reaction post-chromatography purification. The result obtained was an estimated 99.9% of DNA and RNA removal, while having no negative effect on the lentiviral product. The next stage of the downstream was the concentration and formulation. The equipment used was another 0.45 micron TFF and a diafilter with a 300,000 molecular weight cutoff filter. The goals of these stages were to concentrate product and remove contaminants, and to exchange buffers via diafiltration to formulate product for cryopreservation. The novel strategy employed was a second TFF and a diafilter are both novel processes for use in lentiviral separations. The result obtained was product frozen in formulation buffer reduces losses by up to 60%. The next stage was sterilization. The equipment used was a 0.2 micron dead end filter. The goals of this stage were to remove any contaminants that might be left over from the process. The novel strategy employed was a dead end filter has not been described for running a lentiviral production process. The result obtained was an overall product yield of 35 to 40%. Next, we're going to be going into our conclusion section. The aim and method of this project was to enable a price decrease for gene therapy drugs by designing a mostly continuous lentiviral vector production facility in SuperPro that reduce production waste. The results obtained were production of at least 1 times 10 to the 14th virus particles per year at titers of 5.79 times 10 to the 12th virus particles while running at 50% overproduction for contingency. The upstream productivity was 1.6 times 10 to the 13th virus particles per week. The downstream efficiency was 35 to 40% yield. The fixed capital investment was $44.9 million, and the annual operating cost was $17.9 million. Key innovations that we would like to highlight are the unique upstream design, which took advantage of stable producing suspension cell lines, and sequential column chromatography and continuous tangential flow filtration. This work will be concluded with a look towards the future. In the future, we would like to perform profitability analysis with various drugs. This would enable insight into the feasibility of the design facility. We would also like to construct a platform process model to enable direct comparison for cost of production, fixed capital investment, annual production cost, and cost of waste estimation. That concludes our presentation. Thank you for listening.